Bismillahirrahmanirrahim. Assalamu alaikum. How are you, dear students? Dear students, I welcome you once again in Pakistan International School Taif's virtual learning system for the session 2020. And this is biology class for first year, and we are just studying chapter number 4, bioenergetics. And today our topic is Krebs cycle. Before we have studied cellular respiration, its introduction, and then we have seen anaerobic and aerobic respiration. Then we have seen glycolysis, and today the next step we will see Krebs cycle. So in cellular respiration, glycolysis is the process which is common for aerobic and anaerobic respiration. Today we will see Krebs cycle, but before going to Krebs cycle, the very important process by which the molecule of pyruvate is able to perform the Krebs cycle function, that is oxidation of pyruvates. So let's get started. Oxidative decarboxylation of pyruvate, or that is called oxidation of pyruvate, that is done in the mitochondria, but that is happened in matrix of mitochondria, because matrix of mitochondria is having pyruvate dehydrogenase complex, which is having like clusters of 3 enzymes and 5 factors. So collectively that is called a dehydrogenase complex. This pyruvate molecule, to enter mitochondria, it needs a specific protein, transport protein, that is called pyruvate translocase. On entering the mitochondria, pyruvates do not directly participate in Krebs cycle, but it undergo an intermediate phase that is called oxidation of pyruvate, or this is called link reaction. So oxidative decarboxylation of pyruvic acid, that is actually called the link reaction, and this is also the link between the metabolic pathways of glycolysis and citric acid cycle. So this reaction is usually catalyzed by pyruvate dehydrogenase complex. So conversion of pyruvic acid, that is actually the end product of pyruvic acid is the end product of glycolysis, that is done to acetyl coenzyme. So let's go through this process. So for pyruvate oxidation or oxidative decarboxylation of pyruvic acid, that is done in aerobic respiration, that is not happened in anaerobic respiration, because oxygen is needed in the next reactions. So number one, in this reaction, like we can see when we go further, that carbon dioxide is removed in this reaction, as well as nicotine amide adenine dinucleotide, that is reduced to NADH, and the 2 carbon compound becomes acetic acid. Then coenzyme is attached to acetic acid to form acetyl coenzyme. So let's go through this process. So if you can see that this is cytosol of mitochondria, and this is inside of mitochondria. So pyruvate just added into uh, the mitochondria membrane, and that is through the protein that is called pyruvate translocase. Then the next process in which one carbon dioxide is removed out and coenzyme is added, as well as NAD is reduced into NADH plus a proton, and finally we get acetyl coenzyme. So now we will see citric acid cycle. Citric acid cycle is also named as TCA cycle, Krebs cycle, or that is also called citric acid cycle. Because TCA stands for tricarboxylic acids, like the first compound which is formed, that is citric acid, in mitochondria, it is having three carboxylic group inside it. That's why it is called a TCA cycle, or that is given after the name Sir Hans Krebs, who discovered this process. That's why this is named as Krebs cycle also. That is in eukaryotes, all the eukaryotes which are having the proper nucleus, and cycle occurs in mitochondria matrix. From cytosol it goes now into the mitochondrial matrix. So in prokaryotes, CAC occurs in cytosol because prokaryotes don't have membrane bounded organelles. So CAC means citric acid cycle that is going on in the cytosol of the cell. Now we will see the TCA cycle, that is a common metabolic pathway for glucose and fatty acids. Now we will see that citric acid cycle or Krebs cycle. In the first step, pyruvate from glycolysis is degraded to carbon dioxide, which is in that is happened in when there is oxidation of pyruvate, carbon dioxide is removed, and Krebs cycle, carbon dioxide is molecules are also removed, and some ATP is produced, that is almost one ATP molecule is produced. In that, more NADH is formed, and NADH goes to make more ATP in electron transport chain and oxidative phosphorylation, as well as molecule of FAD is also produced, and FADH2, sorry, and GTP is also produced in this reaction. So we will go through these in the next steps. So citric acid cycle is divided into almost 8 steps, which like in 8 steps, which are step by step, we will discuss about that. So energy in the final two carbons are extracted. How the energy is extracted, we will see here. So the first reaction in that is acetyl coenzyme, that is attached to we can see to an oxaloacetate 4 carbon molecule. Okay, 4 carbon molecule, we can have 4 carbon molecule that is formed here, that is OCC, that is OAA, sorry, OAA molecule, which is formed by like a complete cycling, and that is actually recycled again and again to accept acetyl coenzyme, and they're formed after the first compound which is formed, that is citrate, and citrate is the we can say the first stable compound which is formed here, that is also called like we can say it TCA, like tricarboxylic acid. Like it is having at carbon number 1, 2 and 3 three carboxylic groups are attached with that. In from here, when acetyl coenzyme is added by the enzyme citrate synthetase, citrate is formed. This is called synthesis reaction, and in that we can see carboxylic acid, that is called coenzyme A, sorry, that is removed in this reaction. Let's go to the next second one, that is citrate is converted to isocitrate. Before conversion into isocitrate, that is isomerized into cis aconitate, that is isomer of citrate, that is unstable, and that is called dehydration. So hydrogen is water is removed from this reaction. Then the third one is isocitrate reaction. In isocitrate reaction, we can say this is aconitase, this one. And now we can see the complete second reaction, that is the formation of isocitrate. This is isocitrate which is formed here, and water is added in this reaction. That's why it's called hydration reaction. Then we are having the next fourth reaction, that is, sorry, this one, third reaction, that is alpha ketoglutarate is produced. This is the third reaction in which this one alpha ketoglutarate is formed, and in this reaction, that is also called decarboxylation, because carbon dioxide, one molecule of that is removed, and NAD is reduced into NADH. So here oxidation and dehydrogenation, both of the reactions are occurred, and then this reaction is called alpha ketoglutarate, where it is formed here, and one carbon dioxide is removed. So in the next reaction, if you see in reaction number 4, succinyl CoA, that is 4 carbon compound is produced as coenzyme enters into the cycle. So here is succinyl coenzyme A is produced. So what happened in this reaction, when alpha ketoglutarate is gonna change into succinyl coenzyme, from where coenzyme just attached with this one, because coenzyme A is entered in this reaction, and then that is called succinyl coenzyme A. And in this reaction, then one more carbon dioxide is removed, and again NAD, that is called nicotine amide adenine dinucleotide, that is reduced to NADH. So coenzyme re-enters into the cycle, and carbon dioxide is removed as carbon is deoxidized, and NAD is reduced to NADH. So here half of the reaction is completed. So then we will see the next reactions, that citric acid cycle. Now we will see step of 5 to 8. So in that, if we see step number 4, in step number 4, alpha ketoglutarate that we have seen, that is oxidized to 4 carbon compound now, that is called succinyl coenzyme. And now if we just take a look on step number 5, succinyl coenzyme A is then converted into succinate, that is also 4 carbon compound In that reaction, one GTP molecule is produced. This one GTP molecule, it means guanosine triphosphate, that is guanosine triphosphate, that is formed by GDP plus one inorganic phosphate, so that becomes GTP, and then this GTP is uh, just change into, or just phosphorylize ADP into ATP, and then from this GTP we get ATP. So one molecule of ATP is formed is in this reaction indirectly. So then from GTP, this again change into GDP because one phosphate group is just attached with ADP and form ATP. So here one ATP indirectly is formed in this reaction, and then that is changed into succinate. What? Succinyl coenzyme A. Then we will see step number 6. In step number 6, succinate, that is succinic acid or succinate, is converted into fumarate, 4 carbon molecule, and a molecule of FADH2 is produced by FAD. So that FAD is, we can say, reduced, or a proton or electrons are added in this one. So that is also very important cofactor. And now this FAD is reduced into FADH2. Next step is hydration reaction, in which a molecule of water is added to fumarate to form malate. So now malate is formed, that is also 4 carbon compound. In the next reaction, we are having dehydrogenation and oxidation reaction. In that, millet is converted into oxaloacetate, and this oxaloacetate actually is the very important compound which is recycled in this reaction, and again it accepts one more carbon dioxide, and whole of the reaction will move again, and it will repeat the cycle. So in this reaction, NAD, that is again reduced to we know that NADH, and that picks up again acetyl coenzyme, sorry, not carbon dioxide, again acetyl coenzymes, and that will enter into the reaction, and again citrate is formed. So if we see the overview of citric acid cycle, 4 carbon compound, that is oxaloacetate, that condenses into 2 carbon acetyl unit to yield 6 carbon citrate, which is here, that 6 carbon compound. Next step, an isomers of citrate is formed and oxidatively decarboxylized by 5 carbon compound, that is called alpha ketoglutarate. So isomers of citrate, that is called cis acontate, that is intermediate compound, and that is changed into alpha ketoglutarate. Then this alpha ketoglutarate is oxidatively decarboxylated, in which carbon dioxide is removed and yielded to 4 carbon compound, that is succinate. Oxaloacetate is then regenerated from succinate here, and 2 carbon atoms acetyl coenzyme again enter into the cycle. 2 carbon atoms leave the cycle in the form of 2 molecules of carbon dioxide. So 3 hydride ions, like 6 electrons, are transferred into the 3 molecules of NAD, one here, and one here, so we can get 3 of these, and 1 pair of hydrogen atoms that two electrons are transferred to one molecule of FAD, so that will become FADH2. So let's take a look of this Krebs cycle again, like pyruvate this one pyruvate from glycolysis, that is reduced into 2 molecules per glucose. So when uh, after at the end of glycolysis, 1 molecule of glucose is broken down into 2 molecules of pyruvate. So every pyruvate molecule will move and just change into 1 molecule group of that is CoA, acetyl coenzyme A, and then enter into this Krebs cycle. So for one glucose, Krebs cycle has to perform twice, 2 times. So whatever the energy products are formed here, like NADH, ATP, FADH2, we have to just multiply it with 2, because there are 2 pyruvate molecules, that Krebs cycles 2 times it has to be performed. So here you can see oxaloacetate is 4 carbon compound, then it will add acetyl coenzyme A, so it will form 5 carbon compound, then 5 carbon compound isocitrate will change into 4, then 5 carbon compound. When this carbon dioxide is removed here and here, then it will become into 4 carbon compound. And then we can see that at the end we are gonna have oxaloacetate compound, which again just enter into the Krebs cycle and again accept acetyl coenzyme for further reactions. So what is the net we get from Krebs cycle? What is its importance? Like in oxidative process, in the Krebs cycle, we get 3 NADH, 1 FADH2, and 1 GTP. But per glucose it has to be repeated twice. So if we multiply it with twice, we'll get 6 NADH, 2 FADH2, and 2 GTP. And from GTP, like, it is turned into ATP, that is called oxidative phosphorylation. So here that is called substrate level phosphorylation, that GTP is changed into ATP, and that is 2 ATP we will get from here. And ultimately NADH and FADH2 are changed into ATP in a next electron transport chain. Except this, we get 4 carbon dioxide molecules in that. So in respiration we exhale carbon dioxide carbon dioxide. So what are the inputs of Krebs cycle? Krebs cycle consumed 2 acetyl coenzyme or acetyl groups, then it has consumed 2 ADP, like in the form of GDP, and then it is consumed 2 NAD, which is reduced to NADP, and 2 FAD, which are reduced in the form of FADH2. So, then we will see in electron transport chain that how many molecules of energy like ATP we get from electron transport chain, because electron transport chain is the main we can say the main reaction in which the energy is produced. And after all, respiration purpose is to produce energy in the body. That's it, my dear students. I hope you understood this whole topic of Krebs cycle, or how the Krebs cycle is moving on. And next we will continue this to the next topic, that is ETC, or electron transport chain. Wish you all the best. Allah Hafiz.